methods that can be used for that. You can, for example, squeeze the cells through a tiny little pinhole in a metal container, and by the force of shear force of pushing the cells through, that can break the plasma membrane as the cells pass through this kind of little syringe. And then you have broken components on the other side that you can try to begin to separate. You could also use sound, ultrasound waves to break the cells. You can use a mortar and pestle to just, again, break the cells open. There's a variety of ways that you could try to do this. In some experiments down the road, we are going to use detergents that can begin to dissolve the plasma membrane, but in a much more gentle way than if you were to use SDS. SDS is really strong, really dissolves everything, so it's really hard to control. But there are some other detergents that are quite mild, and at small concentrations can just solubilize small portions of the plasma membrane, poking holes or breaking the membrane in such a way that it leaves everything else in an intact way. So once we have this broken cell in a gentle way, we say we have a whole cell lysate that, so the cells have been lysed, so we have a lysate of the entire cell. That's a whole cell lysate. Again, if I want, I can just boil that in SDS and put it on my gel if I want to, but most of the time we're going to try to separate components. One of the most common ways of separating components is by differential centrifugation, which takes advantage of this very simple fact that big things are heavy, so if you put them in a centrifuge, they're going to sediment in the bottom of the tube much faster than tiny little things. That's the principle of the experiment, right? So if you begin to take your whole cell extract and spin in a centrifuge at low speed, the first thing that would come out is whole cells. So if you had some cells that hadn't been broken, that would be the first thing because it's intact cells, right? So you get rid of the intact cells, and then you crank up the speed a little bit more, and the next big thing is probably going to be the nucleus because it's a very large organelle, so the nucleus is going to come down. You crank up a little bit more, you're going to have big chunks of plasma membrane and cytoskeleton. Things like the cytoskeleton that are very large and complex are going to come down. In subsequent, so if you keep repeating the experiment in multiple ways at different speeds of centrifugation, you're going to separate the cell into a variety of components. Depending on the speed that you choose, you're going to bring down onto the pellet different components. So in this experiment, at the low speed, they're not telling us how many G forces they're applied here, but at this particular speed that they selected in this experiment, they broke down whole cells, nuclei, and the cytoskeleton. Why did they do that? Well, maybe they were not interested in that at all, so why do 3 steps when you can just get rid of those 3 things in 1 step? That's just a matter of what it is you want to do. If you want just the nuclei, you would first have to get rid of the cells, whole cells, and then bring down the nuclei, and then everything else. All right? So you have to be wise in how you choose. But by choosing wisely the speed, you can bring down onto the pellet the component that you want. The experiment is not 100%. There's going to be quite a bit of contamination from other components in there. But there's going to be a nice enrichment of components in that pellet. So then what you do is you suck up the supernatant, or you collect it, if that's what you want. And if you want to analyze a pellet, you add some additional buffer, clean buffer, to resuspend your pellet, and now you can do experiments with that. You can do a lot of experiments with that, or you can just simply take that pellet, dissolve it with SDS, and load it onto your gel. Right? Questions? Simple. All right. So let's combine that with an actual experiment with a Western blot. All right. So what I'm going to ask you all to do is to read through this next 2 slides of experiments. We're going to go through them. I'm going to set up the experiment here and explain it. And then I want you to work with your classmates to try to solve these problems just, there's not going to be an Instapoll, just writing down your answers on your handouts, all right? So here's the idea. We're going to take some cells. We're going to break them open gently, and that's going to be our whole cell extract labeled in this gel as W. So we take a sample of whole cell extract, add hot SDS, beta-mercaptoethanol to dissolve everything in there, and run it on the lanes that are labeled W, whole cell extract. Then, to the rest of it, that is not dissolved, we're going to separate on a centrifuge at a speed that should bring down the nuclei. All right? So at low speed, all right? In this experiment, only the nuclei are pelleted in this experiment. So we bring down the nuclei to the pellet, and then we collect the supernatant, we collect the pellet, we dissolve them in a nice buffer, all right? And then we take samples of those, boil them with SDS, and load them onto the gel next to the whole cell extract. So in this gel, we have whole cell extract, pellet, supernatant, whole cell extract, pellet, and supernatant. All right? And I've run multiple gels with those samples, transfer them to membranes, and then those membranes, which are basically replicas of each other, all right? Then I can test, I can query, I can sample with antibodies against different proteins. So in this experiment right here, I took those gels and those membranes and incubated with an antibody against a protein called RCC1. In a separate experiment right here, and they're just put side by side to compare, against a protein called tubulin. We're going to talk a lot about tubulin. We're going to talk a lot about RCC1 down the road. And in this segment of the gel, an experiment against an antibody against histone H3. You all know what histones are. Histones are proteins that bind DNA and to form nucleosomes in the nucleus, right? So we have proteins of markers of the cytoskeleton, which is in the cytoplasm. So tubulin is a cytosolic protein. Histone is a nuclear protein. And then RCC1 is a protein that we are actually trying to learn something about, right? So the experiment tells us that RCC1 is a protein that has been found to be existing in a mutant form, and of course there's a normal wild type form. And they're trying to understand the function of this protein in, I want to make sure I don't give you any more clues than I should. All right, great. Any questions so far? You all understand the experiment? Mm-hmm. All right, so the first set, so these are the questions that I want you to address, which is, why do you see a tubulin band in the W and S lanes, but not in the P lanes? Why do you see a H3 band in the W and P lanes, but not in the S lanes? And then based on those results and everything else in regard to RCC1, what can you conclude about the distribution or fractionation of wild type and mutant RCC1 proteins? So go at it, try to collaborate with your neighbors, try to get answers, and then what I'm going to do is I'm going to try to engage a whole conversation of the entire classroom, all right?